What's up everybody? So in this video we're going to be talking about everything that you need to know regarding the cell membrane structure. So let's just get into it. So picture this, you're a human being, I'm a human being. What are we made of? We are made up of billions and billions of cells, right? Cells have a lot of organelles like the nucleus, the mitochondria, and all that. One thing that all cells have in common is what? The membrane structure. The membrane structure. So membrane is basically the thing that surrounds the cell. If we look at this cell, this would be the membrane. It's the part surrounding the cell. And it doesn't only have to be this membrane surrounding the cell. The nucleus can have a membrane. The mitochondria can have a membrane. So the organelles can also have their own membranes. Now, what is the purpose of a membrane? The purpose of a membrane is two things. One is it provides shape. Without this membrane, the cell would have no shape. All of the organelles would leave out, go to a different place. There would be no structure. There would be no organization, right? So one key function of the cell membrane is to provide a shape, a structure, organization. Now, another one is this, activity with the outside. This membrane can touch other cells and other chemicals outside of the cell and interact with them, okay? And we're going to get into more what this means later. Now, what exactly is the structure of a membrane? What am I talking about when I mention this membrane? How does it look like? If we look real close, what would it look like? I'm going to delete these for now. If we look real close here, what's it going to look like? So let's say we look in here-ish. What we're going to see is something like this here. So we see, I'm showing you a small segment. I'm only showing you this segment, but in reality, this structure will continue on and on both sides until it goes all around and covers the entire cell, right? So I'm showing you only a small section of it, but the structure repeats and repeats. So if you understand one segment, you understand how the rest of the thing looks like. So what is going on here? We can see that this membrane is made up of a repeating unit. This one looks the same as this one, which looks the same as this one, and this one, and this one, and it repeats on and on and on. And we can see there's two layers. We have one layer here on the top and one layer right below. The layer below is in mirror image with the one on top. In other words, it is flipped around. They're not facing the same direction. And we're going to talk exactly later about why this is. Why is the membrane like this, and why isn't the bottom layer facing the same direction as the top layer? Why is it? Don't worry, we'll get to that later. Now, to understand this, know that this side, so over here, is the inside of the cell, right? Because if we're looking here at the membrane, here would be the inside and here would be the outside. So on my picture, here is the inside of the cell and here's the outside, okay? Now, what is this little unit that this membrane is made of? What do we call it? We call it a phospholipid, and we're going to see exactly why it's called a phospholipid just now. Oh yeah, by the way, you need to know this. A membrane is approximately 10 nanometers wide, 10 nanometers. It can vary. It can be 7 to 12 or so. It can vary. So, but around 10 nanometers. This is important because I've seen it come up on some multiple choice questions. So now coming back to this, why is this phospholipid, or this little structure here, called a phospholipid? So a phospholipid, I made it big here, is slightly more complicated than what I made it seem here. It has also this green part also. Okay, I didn't put it here, but it actually looks like this, a little bit more complicated. Don't worry about this word, we're going to come to this one just now. Now, a phospholipid is made up of three parts: a phosphate head, a glycerol, which is the green thing, I'm color coding them, and then a fatty acid, two fatty acid tails. Here's one tail, here's another tail. So this makes sense as to why it's called a phospholipid. Phospho refers to the phosphate head that it has, and lipid refers to the fatty acid tail, because a fatty acid tail, fatty means lipid, right? Fatty means lipid. Lipid is a scientific way of saying fatty. So that's why it's called a phospholipid, because it's got a phosphate group and it's got a lipid, two lipid tails. Now, this structure is two-layered, right? We've got one layer here and one layer here of phospholipid. So we call this structure a phospholipid bilayer, because we've got a bilayer structure. Bi is the prefix for two. It means two, right? Like bicycle, which has two wheels, or bipolar when you have two contrasting emotions. So bilayer or phospholipid bilayer means two layers of phospholipid. So that's real simple, right? Now my question is, why does this structure line up like this? Why on earth does it line up like this? So it's going to make sense real good now. Let me show you. To understand why it lines up like this, I need to show you something. The phosphate head is something called hydrophilic. Hydrophilic means this. Hydro means water, philic means loving. So you can think of the phosphate group as a water loving. It loves water. It loves to be inside water. Whereas this fatty acid tail is hydrophobic, water hating. When you have a phobia, right, you can have a fear of heights, a fear of holes, a fear of spiders, right? So in this case, it's just a fear of water. So the fatty acid tails hate to be in water, whereas the phosphate groups love to be in water. Now, when a molecule has both a water-loving and a water-hating part, we call it amphiphatic. So a phospholipid is amphiphatic since it both loves and hates water. You can ignore this doesn't matter. Yeah, so it's both water-loving and water-hating. So why does this happen? You can think about it. You know that the inside of a cell has a cytoplasm. A cytoplasm is made up of what? Water, mainly water. So it's like a jelly-like substance made of water, which organelles float in. So there's water. Now the outside of the cell is also, guess what? Water. Because we know in reality, humans are like 70% water. That's because our inside of our cells is made of water, and so is the outside. So how does this structure make sense then? This makes sense because if I told you this right here is the inside of the cell, and this right here is the outside of the cell, and this is the membrane separating the inside from the outside, then think about it. This way, the phospholipid heads, the phosphate heads, which are hydrophilic, are facing the water. They don't mind that. They actually love that. Whereas on the outside, which is also water, the phosphate heads are facing the water, and that's great because they love water. And then these fatty acid tails, which hates water, get to face together where there's no water, right? Because when they line up like this, all the water is on the inside and the outside of the cell. There is no water in between these two layers here. And that way, these fatty acid, which are hydrophobic, afraid of water, are completely happy. Right? So that makes sense. Now we know why this happens. And I can try and clarify this a little bit just now. We're going to try and clarify this a little bit just more. So bear in mind, this is something that a lot of people don't actually realize, or they probably know it, but they're not really constantly thinking about it. But I like to constantly remind people that a cell is actually 3D, right? Here I showed you a nice two-dimensional structure. I want you to try and start imagining how it looks like in 3D, because it's not that important for your exam questions, but it's just good to really understand what's going on here. So remember, here I have a picture. A cell is 3D, like a ball like this. And we have a cell membrane surrounding the entire thing, right? It's a membrane. It's like a tennis ball. The yellow part is surrounding everything, like the cell membrane. Now, when we remove the cell membrane, what do we see? We would see cytoplasm, which is basically water, right? And all of these organelles floating inside it freely. Now, what is this picture then? What are we looking at here? What we're looking at here is basically, if you consider this 3D cell, we're basically cutting it open and looking from the top. So we're cutting off the top half and looking at this cell from the top. Therefore, it looks two-dimensional. So we're looking at a cell. We cut it open, sliced it in half, and we're looking from the top. And now that's everything we can see. So I want you to remember that cells are, in reality, three-dimensional, okay? So in reality, when I show you the membrane, it's going to look slightly more complicated than this because I'm showing you a two-dimensional membrane, a 2D membrane, because that's what we would see if we slice the cell open and look at it from the top. How would it look like from a three-dimensional perspective? Let me show you. This is how. So if we imagine, let's go back here, we're looking at like this part of the cell, right? This whole membrane part. And we slice a piece of membrane out, it would look something like this, right? Because this membrane would continue all around the entire cell. So I just want you to get a good perspective of what it looks like 3D, because I only showed you like a small segment like this without showing you 3D. Now this is how it will look like in terms of three-dimensional. So I hope this picture is a bit more clear, okay? Not too important, but I like to emphasize that it is 3D. So now, it's not all. I showed you now this stuff, but it gets a bit more complicated. I was leaving out some things. I was ignoring some things. Now I'm going to reveal the full details, because now you know the basics. Let's add some more details. Oh yeah, by the way, here was a nice picture that if you don't get it, it can help you a little bit. So if you can imagine if we put a phospholipid inside water, what's it gonna do? We know the head loves water and the tail hates water, so it's gonna go like this, right? The water loving part will go in the water, and the water hating part will go out of the water. And so when you have a whole chain of these, they would kind of go like this. Now pretend that there is no air. So pretend this container is so tightly sealed that there's no air, there's just water. What's going to happen then? Then we will have one of two things happen. Either these phospholipids will arrange themselves in a sphere, again a 3D ball, where all the fatty acids will point inwards where there's no water, and all the phosphates will point outwards where there's water, right? Because they love water. So we call this a micelle because it's got a single layer. So other than this micelle, what can happen? A phospholipid bilayer can form, right? Which is what we just saw for the membrane. So we can have basically two layers that form like this. So you can imagine this is the membrane of the cell, this is the inside of the cell, this is the outside of the cell, right? So the inside of the cell has some water, which is why these phosphates are facing inwards. In here could be the nucleus and all these kinds of things. So I'm just showing you a very simplified diagram and why it forms this kind of structure. So now let's add some more details. Look at that love box. So now we're adding a bit more detail. We're looking at it from a three-dimensional perspective because it's more accurate, and we're adding some things. Notice we don't only have this phospholipid bilayer anymore. There's some other things going on. So what's going on here? So the first thing I want to say is this model, what we're seeing here is a theory, right? And we call this theory of the cell membrane the fluid Mosaic model. So this is the currently accepted or the current theory of how the cell membrane looks like. The fluid Mosaic model. The fluid Mosaic model. So what's going on here? So what extra can we label? Let's first start with these blue things. So notice how we have some blue things on top of the membrane and some blue things that spans entirely through the membrane. So these have different names. So we call these blue ones that span all the way through the membrane integral proteins, because they are proteins. They're proteins, so they're made by transcription and translation, and you'll learn about that later. But these are proteins. These span the entire length of the membrane, so we call them integral, because integral means to be inside, to be integrated into a society, to be integrated into the membrane. So these are integral proteins. They are inside the membrane. So specifically, you don't need to know about this for now, but this one right here is a channel protein. It's a protein that it's like a door. It allows molecules and things to go in and out of the cell with just by flowing in and out freely. This one right here is a carrier protein. It carries things from the inside to the outside and from the outside to the inside, but it requires energy in the form of ATP. Now don't worry about this. You just need to know that these are integral proteins. We're going to learn more about these in the the following videos. So what's next? What about these that don't span the entire length of the membrane? What do we call these? We call these peripheral proteins because they are located on the periphery, the outside of the membrane, not on the inside. Cool. What's next? What next? So notice we have some of these peripheral proteins that have this carbohydrate chain on it. Let me put it here. It's got a carbohydrate chain on it. Right? So this one here there's no protein, but there's a carbohydrate chain. But this one has a protein with a carbohydrate chain attached. What do we call this thing? We call this whole thing a glycoprotein. So a glycoprotein is any kind of molecule, any kind of protein that has a carbohydrate chain attached to it. So what's the purpose of this glycoprotein? It has three main purposes. Let's start with the easy one, adhesion. The carbohydrate chain on this glycoprotein, is like a little, it's like little glue. It's like glue. It helps stick to other cells. When this cell moves around and comes into contact with other cells, it sticks to them and they kind of glue together. So one of the functions is adhesion or sticking. That's what adhesion means. Secondly, communication. You can think of it like a little a phone. It's like a little phone or like a little hand. It can grab on and communicate or shake hands with other cells, communicate and things like that. So it's used for communicating. Now, it's also a hormone binding site. So we know your cells can make hormones and send them to other cells. And hormones are little molecules that cause your cells to do something. It's like little messengers. So these hormones could bind here, and this could activate your cell to do certain things. So these are the three, you don't need to know much about that. You just need to know that they have these three basic functions: adhesion, communication, and it serves as a binding site for little molecules called hormones, which ultimately have a function in affecting or manipulating the cell and what it does. Let me label this. So this one right here is a glycoprotein. Again, emphasizing glycoprotein is a peripheral protein with a carbohydrate chain attached. Now, one last molecule is this one here. Maybe you haven't noticed it. These four ring structures, like a hexagon, 4 hexagons put together. We call this one cholesterol. Cholesterol has a huge importance. Its purpose is fluidity of the membrane. So normally the membrane it has a certain consistency. So it's either pretty fluidity or pretty rigid. Now, once it gets too fluid or too rigid, this cholesterol will control that. So for example, this cholesterol, when these phospholipids start binding together really tightly, this cholesterol will influence and separate them so that they don't become too rigid or too tight. Now, at the same time, these phospholipids sometimes can move from this layer to this layer or this one to here. They can move around. They're not stuck there. Bear that in mind. They can move around. Now, if they move too much, then that means that the membrane is very weak, because it's it's like water, right? It becomes very weak. Now, this cholesterol can basically stop this moving. It can control this moving to keep it more rigid. So basically, cholesterol has a purpose in controlling the fluidity of the membrane, making it more rigid when it needs to, or making it less rigid when it needs to. So it has a very important function, making the membrane the right consistency, the right amount of fluid. So that's it regarding the structures and all that. We just got to cover one last thing here. So this is the currently accepted model. It's called the fluid mosaic model. The very first model, so if we go back to 1925, was this model. This is all there was. They only knew about the phospholipid bilayer. Now, 10 years later, in 1935, a guy called two guys, Davson and Danieli, made this model. They believed that the proteins were sandwiched on top of the phospholipid bilayer, kind of like this. So this was later proved wrong by a process called freeze etching, in which they basically showed, using this process, that no, proteins are not sandwiched on top of the bilayer, but rather located inside, which brings us to the Singer-Nicholson model, which was proposed in 1972, which shows that proteins are not sandwiched on top like a sandwich, but they're located in between, inside the membrane. And the method that proved that was called freeze etching. It's basically they sliced open the cell using a freezing machine, and being able to do that, they were able to look at the membrane and see that there was proteins inside. But don't worry, I'll make you don't need to know this method that much, but I just know that this method proved that this method was wrong and proved this method right. This is the currently accepted model, Singer-Nicholson model, also known as the fluid mosaic model, because it's fluidy and it's a mosaic. It's made up of all these different structures, which is like what mosaic means. So that's it for membrane structure. That's all the things you got to know. Just look over it carefully, draw it a few times, and that's it. That's all you need to know.